in tendon regeneration, in bone regeneration, et cetera. So that's not really novel, although we are trying to understand how these cells are working and the efficacy of these cells. But a research project that we focused on quite a bit in the last couple of years was a collaboration between our team at the Orthopedic Research Center and another collaborating lab. Dr. Steve Dow and I collaborate quite a bit on immune and regenerative therapies. And so we co-mentored together Dr. Lynn Pezzanite, who just finished her PhD about 1.5 years ago. So this is largely the work of Lynn's PhD. And one of the things we wanted to look at was the effects of mesenchymal stem cells on infection, because there's been a lot of work in the last decade or so, just in rodents and rabbits, et cetera. So showing that they're great potential to help treat infection. And so just again, a little review of regenerative medicine and musculoskeletal repair. We're trying to figure out the effects of these stem cells and osteochondral defects and osteoarthritis, in bone repair and in tendon repair. So we know that it helps. We're trying to figure out how to utilize these cells for all the musculoskeletal traumas that have just been spoken about this morning. So we have come a long way in trying to understand in our own understanding of how MSCs work. Okay, and so you'll just to kind of a little bit about these cells and their nomenclature. Some people call them stem cells, some people call them mesenchymal stem cells, some people call them progenitor stem cells. But for the sake of this talk, I'm just going to call them stem cells because it's a shorter word. But there's many different terms for these cells because we're still not sure exactly their lineage. So mesenchymal stem cells do many things. We used to think that they became incorporated into the tissue that we put them in, and actually that tissue regenerated because those cells became part of that tissue. And our work over the last decade or so or longer has shown that they actually don't, that's not how they heal. They have many effects on tissue. Those effects are immunomodulatory. So these cells release factors that actually have a fairly strong effect on the immune system. So we call them immunomodulatory. And when you read some of the literature these days now, they'll talk about that term a lot, immunomodulatory. So they decrease or dampen the immune response, which is very important in many things such as osteoarthritis, of course, an infection. So how do these cells affect the immune system? I think many people know, there's different types of white cells, okay? So T cells, T regulatory cells, dendritic cells, natural killer cells, all these cells are within the immune system, and they're considered within the realm of white cells, but they all do many different functions. And so we now know that osteoarthritis and the changes that we see in the joint is not really, it's inflammatory, but it's very much at a low grade. And so we know these cells actually really affect that process when we put them into the joint. They also change the biology of the joint. So in a normal joint, you have things called macrophages. Monocytes turn into macrophages. And in a happy, normal joint, they release things and kind of maintain the homeostasis of the joint. When that goes awry and you have inflammation in the joint, those macrophages then they go into a different form. So easily considered M2, so happy macrophages, M1, unhappy macrophages. And so in that process of osteoarthritis, these cells actually change the environment of the joint, and they make the joint continue to be inflamed. And so we know now in osteoarthritis, we've been looking for a long time. We, as in the world, has been looking for a long time. And now we know that the immunomodulation that occurs in the joint is very important to the progression of osteoarthritis. So one of the things that the Orthopedic Research Center has looked at over the years has been the efficacy of these cells, but we've used often and frequently the cells from the horse's own body. So that's called autologous. That's a tough process because you have to pull the bone marrow or you have to pull the fat. You have to culture each horse's cells out. It's time consuming. It puts the horse under duress because you have to take those tissues, and then you have to grow them up in culture. Wouldn't it be better if we could just take these cells off the shelf from a normal healthy animal and have the shelf treatment? So that's been the focus in the last several years, is what are the differences between autologous and allogeneic? So this is our PhD student, Dr. Amy Colbath, who's now a surgeon at Cornell. And this is just some in vitro assays. I'll just briefly explain, where we look at one of the things that in the cell culture assay that we look at is how these cells suppress those white cells, okay? And so we own cells, different horses' cells can decrease the proliferation of white cells. So the white cells would be from the horse, either and tested against the horse's own stem cells or a different horse's stem cells. We can see that they really suppress inflammation based on these red arrows. And another rats and rabbits, that these cells have very good anti-bacterial effects. And so this is some data from Dr. Dow's lab, where further to having these cells be antibacterial, if you inflame stem cells in a culture, they actually make more anti-inflammatory and antibacterial proteins. So that was first studied again in rodents. And this is just a progression of when you see this light up, this is just a wound that has bacteria in it. When you combine the wound and treat that rodent with antibiotics, not too much different. Stem cells, you decrease it a little bit. If you put stem cells with antibiotics, maybe a little smaller. If you activate those stem cells and you combine them with antibiotics, you really increase the response of the stem cells and decrease infection and sometimes eliminate it. So that was pretty exciting data early on to show that stem cells that are activated and then combined with antibiotics can really decrease infection to a much greater way. That's also shown here. The green here is white cells. When you put stem cells in there, you decrease a little bit. So green is live, bacteria, red is dead. But when you activate those stem cells and you put those against the white cells with bacteria, you actually really decrease infection. So again, some early data to show that that stem cells that are activated and combined with antibiotic. I'm not sure. Thanks. Thanks, Hiva. Okay, so just some more data showing that Staph aureus bacteria, which is often what we deal with in our horses when they get infections in their joints, is often a Staph aureus bacteria, and that's a very hard infections to treat. So that was looked at again in culture. When you activate stem cells in different ways with different mediators that make them, the stem cells, if you will, produce more antibacterial proteins, certain chemicals, PIC, so poly-IC, actually had a very suppressive effect on bacteria. So I'll just move that along, showing that PIC stimulated stem cells decrease bacterial contamination, bacterial colonies in the dish, and decrease biofilm. And I think we'll talk about this in a little bit later lecture, but biofilm is something that we really battle in our equine infections when we have wounds that are open and wounds and infections around plates, right? Super hard to combat because those biofilms that form. So this was exciting. And then we did a few more in vitro culture where we took these activated stem cells and we combined them with antibiotics, and really, so more red means more dead, and showed again continuously that those cells were able to combat infection. So we then took that and decided we really wanted to test that in the horse, because this was a really important finding based on, you know, all the in vitro and small animal data. So I'm going to show you a project that we did. This part of the screen is gone for some reason. I think they're trying to figure it out there, but I'll hopefully be able to walk you through this project, and a project that's leading to some clinical trials that we're starting now. So we wanted to take these activated stem cells, combine them with antibiotics, and we tested this in a horse model. Okay, so this was our design. We took 8 experimental horses and we combined them into 2 groups. We took 4 horses that were control horses and 4 horses that were treated horses. And we put bacteria, Staph aureus, that was highly drug resistant, into the tibiotarsal joints. So 4 horses got staph bacteria. All 8 horses got the staph bacteria. 4 horses got treated with vancomycin. So it was highly resistant bacteria. However, it was sensitive to vancomycin. So we put vancomycin in those joints after 24 hours, after those joints received highly drug resistant staph. And then the other 4 horses got activated stem cells combined with vancomycin. Took those horses out for about 14 days. Some horses actually could not make it that long, and I'll explain a little bit later. But these are the anti-mortem collection data. We looked at ultrasound, we looked at radiographs, we looked at MRI, we looked at pain scoring, and then scored all that and quantified the outcome data. So this is the differences in pain and inflammation in those horses. The horses that were control, so control is in red, blue is the treatment horses. So control means, again, that those horses that had staph bacteria got vancomycin, so that's how we would normally treat those horses, or they got vancomycin combined with activated MSCs. So very huge differences in pain scores. And in the control horses, actually, we had to stop those horses at day 7. The control horses, all of the treated horses that got vancomycin combined with antibiotics were able to make it out to the 14-day mark in a very low grade pain score. This is the synovial fluid from those. So this is a synovial fluid from a joint, again, that had staph, but then got treated with vancomycin. So you can see that's a bacterial culture plate. All horses that got activated stem cells combined with vancomycin, this is the culture plate from the synovial fluid of those horses. So it pretty much eliminated in all of those 4 horses, all of the sepsis that was associated with infection. So pain scoring was significantly better, quantitative cultures, and the biomarkers also within the cell and within the joints that received activated stem cells and antibiotics significantly improved. This was an example. So I always knew every horse's synovial fluid when it came up to the lab. I always knew which one got the stem cells with vancomycin compared to the antibiotic alone, because this is what the control looked like. This is what the activated stem cells combined with antibiotic looked like. And that was uniformly across the whole study. So synovial fluid analysis was significantly improved at the time of the scores for macroscopic scoring was significantly improved. Those horses that had activated stem cells combined with vancomycin compared to the vancomycin alone. These were the contralateral joints that did not have bacteria. Okay, so exciting. This was a very exciting finding for us because these activated stem cells, they're easy to activate in culture. They're allogeneic, so we can take them off the shelf and activate them. And it eliminated very resistant Staphylococcus septic arthritis. So we are starting clinical trials now of 2 cases so far that had fairly horrible bacterial infections in their joints. Both horses, again, it's an N of 2, have responded to being able to completely clear it. So we can deliver some of this data to you all in the next year or so. So a fairly expansive team that has been part of this work. And this is the Orthopedic Research Center within the Translational Medicine Institute at Colorado State. Just a few more slides. And, so that was the big novelty of this treatment that we're studying now. There's some really other important, I think, ideas out there with novel applications of stem cells. Obviously, we all deal with laminitis in a often negative way. And we think that cells, not necessarily activated stem cells, but there are some preliminary, very pilot work that seems to show potentially that stem cells may have a helpful effect on horses with chronic laminitis. Obviously, we need those hoof tissues to grow faster and healthier in those horses. And so that's an area of potential study that's out there, obviously novel, but could be a really great supplement to these adjunct of treatment to these horses that often we still don't know how to treat very well. From that study, in this study that I just showed in this paper here, they combined MSCs with platelet-rich plasma. And then this is an example of a venogram over from day 0, obviously infiltration of blood vessels, and then day 21 after treatment, 51 and day 60, where the blood supply has been reestablished. It's just a case series, very small. Again, so you can't really conclude anything from that. They also changed the biomechanics of the feet in this study, but I do think treatment to these horses that we all struggle with. And then lastly, this is a paper of, just that lists some of the potential of MSCs, novel things, novel implications of these cells outside the musculoskeletal days of treating corneal ulcers, recurrent uveitis, and keratitis. We do have an ophthalmology team at CSU utilizing these cells to try to treat recurrent uveitis and have had some pretty exciting results. We also work with reproductive, our repro team. And so certainly there's a lot of exciting work that's out there currently with using MSC and extracellular vesicles within MSCs to treat things like endometritis, endometriosis, and many various reformation of the uterus. Equine asthma, a lot of immunomodulation that needs to happen in those horses. So that's an exciting area of study. And finally, the integumentary system, wounds, decubital ulcer, etc., that are chronic, that need growth factors and paracrine signaling factors. That is an aspect that these cells may be able to contribute to. So that's all I have. This is our new Translational Medicine Institute, collaborative research. That's where magic happens. So I tried in my best way to figure out how to say thank you. So Thank you to everyone and the organizers. Appreciate it.
No,
We apologize for the mishap. So this is the end of our last session. We're gonna have a moment now I'd like to welcome the CEO of the Jockey Club of Saudi Arabia, Mr. Ziad Al Mughran, who's here on behalf of His Royal Highness